Well, thank you very much for the invitation. It's always a pleasure to come back home, and I feel really honored to be here with this prominent audience and also relevant speakers. I have to change my picture. I have to update the picture. This is the things that I have to, the first thing I have to do for the next time. My talk is going to be about how risk factors for Alzheimer's disease contribute to the pathology of this disease. We are trying to do this in the lab with the known risk factors from a perspective that is coming from the clinic to the basic research and back again to the clinic. And all of you working in Alzheimer's disease recognize this picture, right? This is the amyloid cascade hypothesis that was formulated early in the 90s, by which accumulation of A-beta in the brain induced a cascade of events, including tau phosphorylation, that is leading to dementia. When I try to make an history about how this hypothesis was birthed, start in the late 80s, and then the discovery of the mutations in the early 90s were, let's say, the final proof. There are families that overexpress, or they have mutations, or they have duplications in the APP gene, several different mutations that have Alzheimer's disease. There is another gene that contributes to A-beta formation, which is presenilin. So at that time, we thought we knew what Alzheimer's disease was. By the way, at the same time, the main risk factor for the disease, the presence of ApoE4, was also discovered. If you look about these genetics, what we found is right now we have about 32 APP mutations in the world. We have about 250 presenilin mutations. So we have actually several thousand patients in the world that are carrying mutations. In contrast, we have a large population that are carrying the risk factors for Alzheimer's disease, the most risk factor for Alzheimer's disease. And this is striking, since the effort that has been done in research in these 25 years has been tremendous for the familiar forms, but very few or almost absent for the sporadic forms. The mechanism of A-beta toxicity, I could make several slides because there has been abundant. And actually, this is not easy to see in other disorders. We know very well when a toxin is impairing some mechanism in the neuron, what is the primary target. But in the case of A-beta, it's unknown. So let's say that the mechanisms are multiple. When I started in Alzheimer's disease 20 years ago, we were told that the fibrils were toxic, and then a few years later, we were told that oligomers are toxic. A few years later, that the protofibrils were toxic. Later on was soluble oligomers. 2010, we were told they were the dimers. If you see this progression, you can see that is almost every 4 or 5 years we have a new entity of A-beta that is toxic. So I am waiting for still that something else is coming on. And usually this is a very provocative slide that make a lot of controversy in the people working with A-beta. But this is very relevant because we have been treating patients for the last 10 years against A-beta, different approaches. And we don't know which is the good approach. And Professor Rosso yesterday told us this slide from Mangaliasi and collaborators, where in 2010 there were, let's say, dozens of A-beta, anti-A-beta strategies, and now is very, very few left in the pipeline. So what we know about Alzheimer's disease? We know that Alzheimer's disease, we have several families that carry mutations that have a familiar form. We don't know this familiar form is the same as the sporadic form, but we know that several risk factors contribute somehow to the pathogenesis of the disease. So the sporadic Alzheimer's disease is a combination of genetic risk and environmental risk. About environmental risk is very well described from the 80s already in these big epidemiological studies, that we have factors contribute to the pathogenesis like diabetes, obesity, hypercholesterolemia, and also other factors that they are riskier, so they are protective factors. About genetics, in the last years, we have been hundreds of new studies with increasing numbers of samples, and found a lot of proteins, a lot of genes involved in the disease. You see there are right now almost 700 genes that have been associated with the disease. We are talking a very heterogenic disease. What is telling us? If you put this in a cluster, you will find that actually those genes are related with 5 mechanisms. They are related to oxidative stress, with glucose metabolism, cholesterol metabolism, inflammation, and recycling of vesicles. And this is the same that the epidemiological studies were telling us. They were telling us that oxidative stress, insulin resistant, cholesterol are risk factors for the disease. So epidemiology and genetics, they are shaking hands. They came to the same conclusion. The problem now is to know how this is contributing to the pathogenesis of Alzheimer's disease. So I was lucky to collaborate with John Hardy, who was one of the initiators of the amyloid hypothesis, to reformulate this last year, saying that we have a number of pathways to Alzheimer's disease that, through neuronal misfunction, cause tau and A-beta accumulation. Today, I'm going to tell you one of these. My lab is centered in several of these risk factors, and I'm going to tell you what we have reported and what we are doing right now about impaired cholesterol metabolism and the relation with inflammation, with memory, with renin-angiotensin system, which is hypertension, another risk factor, and with glucose metabolism. So how hypercholesterolemia, how high serum cholesterol contribute to Alzheimer's disease? This has been a big question for many, many years, because cholesterol, the peripheric cholesterol, doesn't pass to the brain. It's impermeable. The BBB is impermeable to cholesterol. And that was shown not so many years ago by a Swedish professor, Professor Ingemar Björkhem, who, to prove that cholesterol doesn't pass to the brain, inject himself radioactive cholesterol and then scan his brain. We don't do this anymore in Karolinska. But Professor Björkhem is still collaborating, is still active in the lab. Well, he also discovered it is actually some metabolites of cholesterol, hydroxycholesterols, that they are permeable. And these metabolites, the most important is the 27-hydroxycholesterol. 27-hydroxycholesterol is produced by hydroxylation of cholesterol by this enzyme, CYP27, and also there is another enzyme that is degrading. 27 is permeable to the brain. So we have more cholesterol in the serum, we will have more 27, and we will have more 27 in the brain. On the other hand, the cholesterol that is produced in our brain by astrocytes is going to be hydroxylized by another enzyme, CYP46, and the resultant oxysterol, 24S hydroxycholesterol, is going to be put out of the brain to the periphery. So this is the feedback mechanism between the two cholesterols, that they are actually in relation. The first study that we did about high cholesterol in blood and memory performance was back in 2009, when we showed that high-fat diet decreased ARC expression in brain. What is ARC? ARC is a protein that is very important, probably the most important protein for memory consolidation. So when the spines are trying to form a new synapses, to form the synapses, to consolidate this contact, you need a change in the cytoskeleton. This change is governed by ARC. And ARC can be expressed as an early gene by two different signaling pathways. It's mediated, the expression of ARC. One is MDA receptor, and the other one is BDNF. And what we show in this study is actually high-fat diet, reducing NMDA receptor signaling in the brain, result in less ARC. By the way, ARC is diminished in Alzheimer's disease brains. 25-hydroxycholesterol, when we treat two primary neurons, has the same effect. So you see a complete depletion of ARC and a very reduction of NMDA receptor signaling. Recently, we have developed an animal model in which we have knockout the enzyme that convert cholesterol into 27-hydroxycholesterol. It's a CYP27 knockout. This animal, we feed the animal with high cholesterol diet, and as you can see, the wild type, they lose ARC, and they have, is the red line, is worse performance in memory tests. But when the knockout animals are subjected to the same cholesterol diet, they don't have changes in ARC, and they don't have changes in memory. So they are fat, but they are very happy, and they don't have any memory problems. So actually, the responsible for the memory problems from the serum, the high cholesterol serum, was the presence of 27-hydroxycholesterol in brain. The second part of our studies was centered in how oxysterols regulate a very important pathway, is the brain renin-angiotensin system. And this is a very intricate, complicated pathway. But when we found this, we were very excited because it's linking up two of the risk factors, serum cholesterol and hypertension, that that contribute to Alzheimer's disease. And this pathway starts with the synthesis of the precursor called angiotensinogen in glial cells, which is secreted, and then converted to angiotensin 1 by renin and to angiotensin 2 by ACE, angiotensin converting enzyme, and so on. And you all know that there are actually epidemiological studies showing that the use of antihypertensive ACE inhibitors and AT1 inhibitors have some positive results in memory and some positive results in the progression of Alzheimer's disease. What we found in those studies was that oxysterols, 27-hydroxycholesterol, but also 24, increased dramatically the activity and the synthesis of angiotensinogen in glial cells and in neuron, and the activity of ACE and AT1 receptor in the brain. Of course, the signaling pathway from the AT1 receptor is very well known. It's an inflammatory pathway, JAK-STAT, that has consequences with vasoconstriction. And we also show that these oxysterols increase JAK-STAT, so increase the pro-inflammatory pathway, and will have consequences eventually for vascular system in the brain. And this mechanism was dependent on a receptor called liver X receptor. And we found this by two different approaches, by treating with an antagonist of LXR, which is 22-hydroxycholesterol, and also by knocking down the receptor itself. When we do these two approaches, the effect of 27 was not seen. So 27 via LXR activate the renin-angiotensin system. What about Alzheimer's disease samples? It was very well described that ACE is enhanced, angiotensin converting enzyme is increased in Alzheimer's disease. We found it in Alzheimer's disease and in patients with MCI, mild cognitive impairment, progressive to Alzheimer's disease, 5 years before getting the diagnosis of Alzheimer's disease. What we found also is that ACE activity and ACE levels correlate very well with 27-hydroxycholesterol levels in blood and in CSF, even in control individuals, which is the black line here. The Alzheimer individuals are the red line. In Alzheimer's disease, we found an accumulation of angiotensinogen in the brain of Alzheimer's disease patients, and we found a dramatic increase of angiotensinogen and angiotensin 1 and 2 in the CSF. We thought at that time that that could be a biomarker for the disease, but unfortunately or fortunately, this is found in almost every neurodegenerative disorder. Because of the JAK-STAT history that I told you and the possible pro-inflammatory effect of 27-hydroxycholesterol, we were focused in screening what is the effect of 27-hydroxycholesterol in pro-inflammatory cytokines and inflammatory markers. And we found this protein, S100A8, or calgranulin A. What is this protein? This is an agonist for RAGE, receptor for advanced glycation end product. And RAGE is well known to be the transporter of A-beta from the periphery to the brain. That was published in Nature 2003. But also is a well-known regulator of BACE, which is beta secretase. The signaling pathways that they are controlled by S100 and RAGE are pro-inflammatory with all these pro-inflammatory cytokines, but also through NGF to the expression of RAGE and BACE. I don't know if you can see here. Otherwise, you have to believe me. High-fat diet increase increase S100A8 in the brain. This is the expression levels. It's a potent increase. And what we found that this S100A8 was found as aggregates, extracellular aggregates that didn't colocalize with any cell marker. The aggregates were not fibrillar, they were kind of oligomeric, but they were quite well identified, very easy to identify, and didn't although you don't see the arrow here, didn't correspond to corpora amyloid acid. So there are aggregates that they are there, they don't bind to A beta in the hippocampus only. S100 is increased by 27-hydroxycholesterol, but also the receptor rate is increased by 27-hydroxycholesterol in these animals on high cholesterol diet. We found the race activation in primary cultures, in vitro cultures with primary cells, treated with 27. We found also the base action, although we could not see this in protein levels, and we found an increase of NF-kappa B and NF-kappa B activation, translocation to the nucleus. For the first time in our studies, those effects that we described for S100 were not depending upon NXR receptor, and actually we don't know which receptor are depending on. We are still looking at it. What we also found is that animal models, models known for Alzheimer's disease, like the APP mice, TG2576 or the Arctic mutant, they have the same big inclusion of S100A8 in the hippocampus that actually appear before the A beta plaques. And this is a theophylline experiment. They don't colocalize with fibrils. These aggregations are not fibrillar, are oligomeric, appear before A beta, and they are in the same places where A beta will appear later. When we play in vitro with these two molecules, recombinant molecules, we found that A beta 42, if you treat astrocytes with A beta 42, will enhance S100A8 expression and production, secretion from the glia. And vice versa, when you treat neurons with S100A8, you are increasing A beta 42 production, but not A beta 40. These are the quantification. It's very clear effect in the primary neurons. So we are actually making this paper, trying to write this paper, saying that it's a positive feedback mechanism between S100A8 production and A beta production. Don't forget that our 27-hydroxycholesterol and our high serum cholesterol in vivo is increasing S100, so it might have consequences also for A beta production. The last part of my talk is the relation between high serum cholesterol and another feature of Alzheimer's disease, that is glucose hypometabolism. There were two papers in the literature from the same group, PET studies showing a relation between cholesterol-related genes or high cholesterol in blood and glucose PET, glucose hypometabolism. All you know that is one of the first features that we can check in Alzheimer's disease, and it has been used, it is still used as even a biomarker for the disease or a diagnostic tool for the disease. And why we think that cholesterol might have an influence on glucose metabolism? Because as I told you before, 27-hydroxycholesterol and high serum cholesterol have a big impact in this pathway, the renin-angiotensin system. And this pathway doesn't stop here. Angiotensin II is going to be converted to different angiotensins, angiotensin 3 by aminopeptidases, aminopeptidase A. Angiotensin 3 will be converted in angiotensin 4 by aminopeptidase N and B. And angiotensin 4 is a well-known substrate for IRAP. What is IRAP? It's called insulin-related aminopeptidase. And IRAP is a very important regulator of many molecules. It's cleaving somatostatin, bradykinin, oxytocin, but also is modulating the activity of this receptor, which is one of the most important glucose receptors in hippocampus. It is the GLUT4. So when you have a high activity of IRAP, then you have a low activity of GLUT4, and vice versa. When you have low activity of IRAP, then you have high activity of GLUT4 and more glucose uptake. Angiotensin 4 is a well-known inhibitor of IRAP. So the first thing that we did to check this hypothesis, the hypothesis that 27-hydroxycholesterol was affecting glucose metabolism in the brain through the RAS system, was, okay, how clinical relevant is this? Because all the patients that have high cholesterol levels are treated with statins or with diet, but all of them are treated, especially old patients. The first hypothesis maybe is a hidden population of patients with high cholesterol metabolism. So they have normal levels of cholesterol. The doctors don't see that they need to be treated, but still they have high cholesterol metabolism and therefore more 27 levels in the blood. So we went to our clinic and select 75 patients in which we have glucose PET, that we have CSF sample and blood samples. We took away all those patients that were treated by hypertensive drugs or by statins, obviously because we don't want to interfere with the RAS system. We want a naive population. Only 21 patients were left, so it's amazing the number of patients that they are treated with these drugs when they have memory complaints. All of these patients have memory complaints, by the way. And then from 21 patients left, half of them, they have high levels, pathological levels of 27-hydroxycholesterol in blood, and the other normal levels. When we correlate this 27 data with glucose PET, so with glucose metabolism, what we found it is a very strong correlation of the levels that the patients have in blood and in CSF of 27-hydroxycholesterol with the glucose metabolism. These are extreme cases. This is a normal patient. A normal patient in terms of 27-hydroxycholesterol, they have less than 1 nanogram per milliliter, 0.5. You see it's a perfect normal in terms of glucose metabolism. And this is a patient in this area with 1.5 27-hydroxycholesterol levels. I mean, this is for us like as Alzheimer's disease patients, right? And we can see this low cholesterol, low glucose metabolism in all the areas that we study in hippocampus, postcingulate, etc. So the question is, okay, let's back to our animal models. Is 27-hydroxycholesterol responsible for this effect on glucose metabolism? And now we have more models in which we overexpress the CYP27 enzyme, so these animals have pathological levels of 27-hydroxycholesterol. We have the knockouts, we have some other models, and of course in vitro assays. And I'm going to show you data proving that our CYP27 animals, they have lower glucose metabolism, they have lower expression of GLUT4, and they have impaired memory. This is a lot of data, but this is our PET, the glucose PET in our animals, the controls, and the CYP27 animals. This is the quantification. You see a tremendous decrease in glucose metabolism in all the areas of the brain. This is the memory performance. This is the quantification of several things in the memory performance, our Morris Water-Maze picture. They saw less ARC, that I showed you in the first part of my talk, and they saw both in cortex and in hippocampus lower levels of GLUT4. So the mechanism, it might be dependent on this, because GLUT4 is impaired. Is it dependent of IRAP? That was the next question. And is those effects mediated by LXR? Because LXR was the dominant for the RAS effect. Here we are treating primary neurons with our positive control, insulin, and measure glucose uptake. As you can see, 27-hydroxycholesterol reduced glucose uptake in our primary neurons. Cortical and hippocampal neurons. At the same time, they are increasing IRAP activity. Remember that IRAP activity was like opposite to GLUT4 activity. So this is the protein levels. This is the IRAP activity when we are treating our neurons with 27-hydroxycholesterol. It's a very high increase, both cortical and hippocampal. And then when we are treating with inhibitors of LXR, the 22-hydroxycholesterol, or when we are knocking down LXR beta, then the effect disappears. So we don't see this effect anymore. For example, knocking down by silencing LXR beta. In our other models, the transgenic animals, we saw that the CYP27 animals, they have an increase in IRAP activity, as we are seeing in the high-fat diet animals and in our in vitro assay, and the knockout animals have a reduced IRAP activity. So these mechanisms are mediated by LXR, by GLUT4, and by IRAP. But how this is working, really? Because the only thing that we know in the literature is that angiotensin 4 was controlling IRAP. What we have found is actually a dual role of this angiotensin. Angiotensin 3 and angiotensin 4 have opposite functions on IRAP, and therefore have opposite functions on glucose uptake. And what actually 27-hydroxycholesterol is doing is enhancing the activity of this enzyme, aminopeptidase N, which degrades angiotensin 4, and then the balance between angiotensin 3 and angiotensin 4 is changed, so we have an inhibition of glucose uptake. This is the data showing this. When we treat with angiotensin 3, recombinant angiotensin 3, we found the same effect as with 27-hydroxycholesterol on glucose uptake and enhanced IRAP activity. When we are treating with angiotensin 4, we have a very good effect on glucose uptake, even better than insulin. When we treat together 27 and angiotensin 4, this is 27 alone, this is 27 plus angiotensin 4, you see that angiotensin 4 is able to counteract the effect a little bit on IRAP activity. What is important to see here is that this partial effect on IRAP is enough to normalize glucose uptake. So this is very promising. If we can find an inhibitor of IRAP that is not so good after all, can inhibit a little bit the effect of 27, this inhibitor will normalize the glucose uptake effect. The effect was dependent on LXR. I mean, that was obvious. This is the effect showing how 27 increased aminopeptidase N. Remember, is the aminopeptidase that is degrading angiotensin 4. So have a very high effect on the levels of these aminopeptidases that disappear when we are knocking down or we are inhibiting LXR receptor. So the mechanism is very clear for us that it's depending on LXR. In vivo, in our animal models, aminopeptidase N is very increased in the CYP27 animals and is decreased in the CYP knockout animals. We abolish the effect also by knocking down aminopeptidase N with silencing RNA. So the mechanism so far that we have found that high serum cholesterol affect the brain is to 27-hydroxycholesterol, first by affecting memory consolidation to a mechanism LXR dependent, NMDA receptor dependent, R dependent. Second, by overactivating RAS, angiotensinogen, also the receptors, and of course the levels of angiotensin 1 and 2, also LXR dependent. We have found that increased inflammation by a mechanism that is dependent of this S100A8, NF-kappa B, base 1 RAGE, the receptor we don't know yet. And finally, through LXR as well, and through an increase of the activity of aminopeptidase N, and then the balance of angiotensin 3 and 4, the activity of IRAP and the activity of GLUT4 is decreasing glucose uptake. So we have an hypothesis. Our hypothesis is that high serum cholesterol through 27 has a lot of effects in important pathways for neurodegeneration, also for Alzheimer's disease. Many individuals have normal cholesterol levels, are not treated with statins, but they have high levels of 27, and these individuals will have these problems. This hidden population that we call have higher risk for active pathways for neurodegeneration. So the doctors say to me, okay, what is the problem? We treat everybody with statins. And then what are the consequences of treating everybody with statins? If the patient has high cholesterol levels, you will normalize cholesterol level, you will neutralize the damage. Perfect. If the patient has normal cholesterol levels and normal cholesterol metabolism, what you are doing is decreasing cholesterol and decreasing cholesterol function. So why to treat these patients? They are normal. And if the patient has normal cholesterol levels, but they have high cholesterol metabolism, then you will further decrease because you have, with the statins, you are doing a depletion of cholesterol synthesis. They are high metabolizers. They will oxidize more cholesterol, they will decrease cholesterol levels, and you will increase damage. And if you go back to the trials of statins in Alzheimer's disease, the prospective trials, one of the side effects that were found is memory complaints. So the people with Alzheimer's disease treated with the statins, some people, they have a higher progression, a faster progression, more memory complaints. So based on this hypothesis, what we are doing now is first we are developing a diagnostic tool. How can doctors check these patients with high 27 levels that they are hidden by the normal cholesterol assays? Second, once we identify these patients, can we develop some drugs? Again, first, the metabolites of cholesterol, CYP27 inhibitors. This will be the perfect drug because you don't need to reach the brain. The enzyme is peripheric. And even you can reach the brain by other approaches like IRAP inhibitors or APN inhibitors. Our theory is that with this approach, you will normalize, you will treat effectively people by which the neurodegenerative process is driven by cholesterol, impaired cholesterol metabolism. So positive effects on condition, normalization of RAS in brain, anti-inflammatory effects, probably reduction of the S100A8 plaques, and also normalization of the glucose hypometabolism. So what is, in our perspective, the vision and the future of the field? I think that we have a lot of information in this, in risk factors, insulin resistance, cholesterol metabolism, antioxidant deficiency, etc. And probably there are some different variants, some different mechanisms that they are resulting in neurodegeneration of Alzheimer type, of on other types, because they are overlapping, as you know. And also we could find early biomarkers for those drivers of the disease. And this is very important, not only to develop new targets, but also to adjust the clinical trials that we are doing now, because we are doing now, as I showed you in the beginning, almost blind. And this is the final, and this is the people I would like to thank. Of course, there are more people involved. This is Ben Bimblad, director of our center, Gunilla, our scientific coordinator, Ingemar Bjørgen, the one who injects cholesterol, radioactive cholesterol himself, is still collaborating with us, Lars-Olof Wallon and Irina Savicheva in the clinic, Laura Mateos, mainly responsible of the first studies, the RAS studies, and the ARC. Mustafa Ismail is the glucose uptake studies. Silvia is our expert in behavior. Maria and Paula, S100, and the rest of the group. And thank you, all of you, for your attention.